So I'm going to go on and talk about CRISPR. Okay, so this is what you're all waiting for, right? So what is it? It's a protein that has two nuclease sites that will cut both strands of DNA. And it's guided to do it by an RNA. So it's a two-component system. It's the so-called Cas9 protein and a RNA that has two components. It's called a guide RNA. And one component is a stretch of nucleotides, generally about 22 in length, that tell the Cas9 protein where in the genome to bind. And then there's a second part of the guide RNA, here colored blue, that binds to Cas9. So what the RNA does is link the enzyme to DNA. One hand of the RNA binds to the enzyme. The second arm tells it where to go on the DNA. And what it does in simple terms is separate the two strands of DNA so that the enzyme can cut both strands, diagrammed here as a scissors. What I'm going to, okay, I mean, I'm not even going to go through how it was derived. This is the only reason for showing this. The RNAs were originally two RNAs. And Jennifer Doudna in this paper combined the two into one. And this is the paper that caused all the patent fights. This is the 2012 science paper that they did it first, but group at the Broad Institute, literally down the street, was the first one to do it in human cells. And there was the base of a huge patent fight, which is still unfolding. So who did it first anyway? Let me go to the movie. Because again, I think the movie will explain it. But again, what you're seeing here is an enzyme that cuts DNA. And it can either be repaired by non-homologous end joining, in which case it will create usually a deletion mutant, getting rid of something, or if you put in an extra piece of DNA that has the sequences of the ends of the cut DNA, you can often insert a foreign sequence of DNA. So let's get the movie on.
Single nucleotide mutations can be introduced into a genome using the CRISPR-Cas9 system. CRISPR evolved as a type of acquired immune system in bacteria to precisely target and cleave invading foreign DNA. It has now been adapted for use in many organisms for genome editing. The engineered system has two components, which can be delivered by transfection of plasmids. One includes a copy of the target sequence from the genome. The target sequence is joined to a sequence called the tracer RNA scaffold. This construct is transcribed from a species-specific promoter. Transcription produces a functional chimeric guide RNA. The tracer RNA portion has palindromic sequences that allow it to base pair with itself and form hairpin loops. In the second construct, a gene encoding the enzyme Cas9 is placed next to a coding sequence for a nuclear localization signal. A species-specific promoter allows this construct to be transcribed into mRNA. Now we have two types of RNA molecules. Another version of this gene editing technique involves introducing the guide RNA and Cas9 mRNA into the cell directly. After translation in the cytoplasm, Cas9 will be targeted via the nuclear localization signal back to the nucleus to do its job. Cas9 is a double-stranded DNA endonuclease with two distinct nuclease active sites. The guide RNA binds to Cas9 with its hairpin scaffold structure. The target sequence, which is about 20 nucleotides, corresponds to the targeted site in the genome. This complex targets the genome by base pairing of the guide RNA with a complementary genomic DNA. Cas9 cleaves both strands of the target DNA in the chromosome. The resulting double-strand break in the specific target sequence is typically repaired by a set of enzymes that ligate the free DNA ends back together in a process known as non-homologous end joining. Typically, a few base pairs are deleted at the site of cleavage because nucleases remove bases from the free DNA ends before the end joining reaction is completed. If the cleavage site is within a gene coding sequence, the deletion of bases will usually cause a frameshift mutation, which would garble the downstream coding sequence. Here it results in a premature stop codon. The frameshift inactivates the targeted gene. Let's go back to the DNA before the cut strands have been resealed. An important refinement of this method of genome editing is the addition of a segment of DNA, typically about 100 nucleotides long, that matches the sequences flanking the cleavage site. When homologous DNA is present, the free ends can be repaired by homologous recombination, or homology-directed repair, using the added DNA segment, which acts as a template. The template can have a specific change that is to be incorporated into the genome. So by appropriate design of the guide RNA to specify the cleavage site, and by the introduction of appropriate sequence alterations in the added homologous DNA segment, exact single-base changes in the target DNA can be produced by this CRISPR-Cas9 system.
Okay. You like it? Questions? Yes.
Can you speak to some of the key factors that drive lack of specificity in the system?
Sure. Let's go back here. I mean, it all depends on base pairing between the RNA and the DNA. And what is standardly depicted here is perfect base pairing. But suppose there's a sequence somewhere else in the genome that differs from this target sequence by only one or two bases. You won't have perfect base pairing, but you may have sufficient base pairing so that the enzyme sometimes goes to the wrong site. And that's what happens. Now, you can say, well, I have the sequence of the whole human genome. But not everyone's genome is the same, and even these predictive techniques don't work. So it's not a perfect system. And the same is true with the talons and the zinc fingers. Now, if you're gene editing an apple, it's not so significant. But if you're gene editing a person, it can be significant. It's a great question. Yes? Yeah.
Can you speak a little bit about the how does chromatin packing and epigenetic factors affect your ability to target?
They do. Obviously, if the gene is not active in a cell, it will be harder for the enzyme to get to. And again, it's not a perfect system. And this is some of the problem. You see, if you're trying to do it in vivo, usually you'll deal with a tissue or cellular tissue in which the gene is active. But if you want to target a stem cell, it may not be. As I said, it's an evolving form, and we don't understand all the variables. Andrew, where are you? Andrew Hack?
I still know how to work this thing here.
Oh, yeah, press the button.
Right. Yeah, hey, I'm Andrew Hack. I was at Editas for the last 4 years. And
That's one of the big gene editing companies.
I would just make two comments on the questions posed. One related to specificity. So this is a field that's very actively under investigation. I think what we have shown and others in the field are that by taking the time, really I'd say the time that only a company can take to optimize the system, you can actually get to gene editing enzymes using CRISPR-Cas9, CRISPR-CPF1, or zinc finger constructs that have no detectable off-target edits in the genome, but that requires some proprietary technologies. I think one thing to bear in mind in the academic research field where these get used as reagents and often are not highly characterized is that in doing experimentation in that context, it is important to really be sure that the effect that you're seeing is due only to on-target effects, because whereas for a company, we could spend a year getting to a place testing hundreds of constructs using different mutant forms of the enzyme, higher specificity forms of the enzyme, things that are proprietary to the company to get to enzymes that have no detectable off-targets, which is important for therapeutics. A lot of what gets done in the academic setting, in fact, doesn't have the luxury or the ability to get to that place. So I think it's worth bearing that in mind, that where the state of the art is today is you can drive these to the kind of specificity that you would want in a therapeutic, but it is good to remember what Harvey points out, which is that I wouldn't take specificity as a given when you look at results that come out of this field. So that's to your specificity question. And this was true, by the way, in the siRNA field when siRNA was first developed and spread like wildfire in a similar way in the research community. People went out and did experiments where they knocked down this gene or that gene and saw amazing things. And then over time, as it emerged that the specificity profile of siRNAs was not as simple as just picking a sequence that looks like it's what you want, that field also had to wrestle with specificity, both in the academic context and also in the therapeutic context. And then remind me of your question again?
Talking about epigenetic effects.
Oh, yeah.
And chromatin packing.
Yeah, so I think a very reasonable question. I think Harvey's comment is a reasonable first approximation of the answer, although when that has been investigated in detail, it's probably more complicated than it appears. And so there are sites that are not accessible to editing for various reasons. Harvey's point about stem cells, I think, is a very important one, which is that you have a whole sets of genes that are chromatinized and modified in a way that makes them less accessible, although we and others have shown successful editing of genes that would be predicted to be repressed and chromatinized. On the flip side, people sometimes struggle at certain sites and try to understand why that is. And it's easy to say it's the chromatin and packing, but I think it's a more complicated exercise. And some of the rules around efficiency and specificity are still not clear, despite a lot of work.
Good.
Yeah.
Thanks.